Welcome to this informative video on the structure and function of the mitochondria. We will learn this topic in two videos. In the first video, we will learn about the structure of the mitochondria, whereas the function of the mitochondria in the second video. Mitochondria, often referred to as the cellular powerhouses, are double-membraned organelles found in most eukaryotic cells. Their unique structure enables them to perform essential functions related to energy production and cellular metabolism. The outer membrane of mitochondria is porous and contains various protein channels called porins. These porins allow the passage of ions, metabolites, and small proteins. The outer membrane also helps protect the organelle from harmful substances in the cytoplasm. The inner membrane is highly impermeable and contains a variety of specialized proteins involved in ATP synthesis and transport. It houses the electron transport chain, which consists of protein complexes that transfer electrons and actively pump protons across the membrane. The inner membrane of mitochondria is folded extensively, creating structures called cristae. These cristae increase the surface area of the inner membrane, providing more space for ATP synthesis and enhancing the efficiency of oxidative phosphorylation. The intermembrane space is the region between the outer and inner membranes. It contains a lower concentration of proteins compared to the matrix, but is rich in enzymes involved in lipid metabolism, including enzymes responsible for breaking down fatty acids. The matrix is the central compartment of the mitochondria enclosed by the inner membrane. It contains a semi-fluid matrix solution that houses various enzymes involved in cellular respiration, including those required for the citric acid cycle or Krebs cycle. The matrix also contains mitochondrial DNA ribosomes and tRNA molecules necessary for protein synthesis within the organelle. Mitochondria possess their own DNA, separate from the nuclear DNA. Mitochondrial DNA is circular and encodes a small number of genes essential for mitochondrial function. These genes primarily code for proteins involved in oxidative phosphorylation and ATP production. Mitochondria are capable of dividing to replicate themselves within the cell. This process, known as mitochondrial fission, involves the constriction and separation of the organelle into two daughter mitochondria. This division is essential for the maintenance and distribution of mitochondria during cell growth and development. Mitochondria can associate and interact with other cellular structures, such as the endoplasmic reticulum and cytoskeleton. These associations facilitate the exchange of lipids, calcium ions, and other molecules between mitochondria and other cellular compartments. The membranes of mitochondria are predominantly composed of phospholipids, similar to other cellular membranes. However, the lipid composition of mitochondrial membranes is unique and plays a role in their function. The inner membrane, in particular, contains a high proportion of cardiolipin, a phospholipid that is important for maintaining membrane integrity and facilitating the activities of various proteins involved in ATP synthesis. The inner mitochondrial membrane houses several protein complexes that are crucial for oxidative phosphorylation and ATP production. These complexes include NADH dehydrogenase or complex 1, succinate dehydrogenase or complex 2, cytochrome BC1 complex or complex 3, cytochrome C oxidase or complex 4, and ATP synthase or complex 5. These complexes work together to transfer electrons and pump protons, creating an electrochemical gradient that drives ATP synthesis. Mitochondria have their own ribosomes, which are smaller than ribosomes found in the cytoplasm. These mitochondrial ribosomes synthesize some of the proteins required for mitochondrial function. The proteins synthesized within mitochondria include those involved in oxidative phosphorylation, as well as other mitochondrial-specific proteins. Mitochondria form a dynamic network within cells, constantly undergoing fusion and fission processes. Fusion allows mitochondria to combine and share contents, including DNA and proteins, while fission results in the division of mitochondria into smaller units. This dynamic nature of the mitochondrial network helps maintain a balance of mitochondrial function and quality control. Mitochondria and the endoplasmic reticulum form close associations called mitochondria-associated ER membranes, or MIAMs. These contact sites allow for the exchange of lipids, calcium ions, and other molecules between the two organelles. MIAMs play important roles in various cellular processes, including lipid metabolism, calcium signaling, and the regulation of mitochondrial dynamics. Mitochondria are inherited maternally in most organisms, meaning they are passed down from the mother to her offspring. However, there can be some variations in mitochondrial inheritance across different species. Once inherited, mitochondria can be distributed throughout the cell by active transport along the cytoskeleton, ensuring a proper distribution of energy-producing organelles. These details highlight the sophisticated design and organization of mitochondria, which are essential for their crucial roles in energy production, metabolism, and various cellular processes.